Hello, everyone. Welcome to my video tutorials for spatial dataset analysis. Today, I'm going to show you how to analyze the Nanostring CosMx non-cancer dataset using the Giotto package. I already showed you how to analyze this dataset using Surat in R, and also analyze this dataset using the ScriptPy package in Python. So Giotto is another R package that can analyze spatial dataset. So I'm making video tutorial for the Giotto package to analyze the Nanostring CosMx lung cancer dataset again. So the dataset I'm using for today's demonstration is the lung-12 sample. If you want to use this dataset for practice, you can go to Nanostring website. You can see here is the link for download the data for lung-12. In my Surat and ScriptPy package, we use the lung-5 repeat 1 for the demonstration. You can see the data size. Lung-12 is 1.1 and lung-5 is 1.5 GB. So lung-12 is a little bit smaller. I tested it on my computer. I can use this dataset to analyze the full images. So the online tutorial also used the lung-12 as the demonstration dataset. So in the first step, we load the lung-12 dataset and prepare the data for creating a Giotto object. We can have a look at the data files in my folder. You can see here is the data for lung-12. We have the cell composition folder, cell labels folder, cell overlay, and cell compartment labels folder. In each folder, we have 28 images. We can click the first one and have a look. You can see in this folder, we have 28 images for the cell composition. It will be the same number of images in other folders. Then we have the CSV file for the expression matrix, the field of view position files, the metadata, and also the TX file. The TX file contains the subcellular transcripts information. So now we can load the data into R. So first we load the TX file to get the coordinates for all the probes. Let's load the data. You can see we have 9 columns in the TX file. We can have a look at the column's name. Let's run. You can see the first column is the field of view position. We have 28 images here. The second column is the cell ID. Then the third and the fourth column holds the information for the X and Y global pixel position. Then after that is the X and the Y pixel position on each file. A column is the Z-plane for detection. Then the target are the gene targets for the probes. And the last column is the cell compartment information. So we have the gene name in the target. We can get all the probes ID from the target. Let's run. You can see in this dataset, they use the 980 probes for the in situ hybridization. You can see the gene name here. Because they also use the negative control probes for in situ hybridization. So we can split the feature ID and the negative probe ID. We know all the negative probes has a name as a negative probe. So we can get all the negative IDs. Let's run. You can see we have 20 negative probes in this dataset. Then the rest probes will be the features ID. We can run. You can see we have 960 probes. For the data analysis, we need to separate the gene expression data from the negative probes data. So we can split the detections for the in situ hybridization data to feature coordinators and also negative coordinators. So we can use the feature ID to get the detections for feature coordinators. Let's run. You can see we have the features coordinators here. The next we can get the negative probes coordinators. We can write again. You can see here is the detection for the negative probes. So we separated the gene IDs and the negative probes. Next, we can note the field of view positions. I showed you we have 28 images in this dataset. So we need to choose the field of view for the analysis. The online tutorial showed you how to analyze the field of view number 2, 3, and 4 together. But today I'm going to select all the field of view and do the demonstration. So we need to set the field of view ID as number 1 to 28. Let's set it. So we are going to analyze the whole image. So it will take time to finish this demonstration. So we know all the data. Now we can create a Giotto object for each field of view. First, we need to create a Giotto object list. Let's create it now. Then, for the following code, we will create an individual Giotto object for each field of view and make a Giotto object list. So you can see first we can get the original image information for each field of view. Then we get the data for the cell segmentation for each field of view. After that, we can get the feature coordinators. After the feature coordinators, we can get the cell central node to provide the spatial location for each cell on each field of view. Then we add the images into the object. So let's run the code we set here to create an individual Giotto object. Let's run. We will create 28 individual Giotto objects and make a Giotto object list. It will take some time to create all the individual Giotto objects. Okay, so it finished to create all the individual objects. You can see now in our Giotto object list, we have 28 objects. So next we can join all the individual objects and create a joint object. So first we get new names for each individual field of view. Let's get the names. And also we need to match the numerical ID for each individual object. I showed you in the TX file, we have the global pixel position for each field of view. So now we can set the position for each individual field of view as the global pixel positions. We set the X position first. Then next, we set the Y position. So now we can use the new names for each individual field view and also use the global position to create a joint Giotto object. We named it as field view join. Let's create the joint object. So we created a joint object. You can see in the data window, we have a Giotto object named as field view joint. We can click this object and have a look at the data structure. You can see we have the expression data here. Then the spatial information. We have the cell metadata. We can have a look. You can see in the cells, we separated the feature ID detection and the negative probes detection. So you can see the RNA for the feature detection and the detection for the negative probes was saved in different data slot. Then we have the feature metadata. It will be the same for the RNA and the negative probes. You see we have 960 feature ID, then 20 negative probes. Down here we have the feature information. You can get all the gene names for the features and also negative probes names here. So that's the data structure for the joint object. Now we can set the image names for the joint object. Later we can use the image names for data visualization. So let's set the image names. So in the next step of analysis, we are going to aggregate the subcellular features. If you want to understand why we want to perform this step analysis, you can have a look at the online tutorial. They have detailed explanation for this step analysis, and also they compared the aggregated features with the original features, and they show the aggregated features are very similar to the original Giotto expression matrix. So first, in this step analysis for feature aggregation, we calculated the feature points overlapped by the polygons. Because we separated the gene expression data from the negative probes detection, so we need to calculate the feature point for both gene and the negative probes. So let's calculate the feature point. First is for the genes, then next is for the negative probes. So after the calculation, we converted the overlap information into a cell by feature expression matrix. And this matrix information will be saved in the Giotto object expression slot. So first we converted the overlap information for the genes. Then we converted the overlap information for the negative probes. Okay, we should save the aggregated subcellular features into the object. We can perform next step analysis. Next step will be step 5. In step 5, we perform data filtering. So before we filter the data, we can use the filter distribution function to look at the detected features in each cell. Again, we are going to look at the RNA and also the negative probes. So we can have a look at the distribution for RNA first. Let's run. You can see we generated a histo figure here. You can see the total features in each cell. On the axis are the cell numbers that have the same total features. Next, we can have a look at the filter distribution in negative probes. We can run. You can see the total features cell for the negative probes are below 10. So now we can set a threshold to filter the object. For the filtering, we only filter the RNA data. We are going to use the expression threshold as 1. Minimum cell numbers for feature detected as 5, and also the minimum features detected in each cell as 5. So this is the same threshold that have been used for online tutorial. We can use the threshold we set here to filter the data. Let's run. So after data filtration, we can perform next step analysis, data normalization. So the online tutorial used the 2 steps of data normalization. So the first step is the standard method of normalization. It is a log normalization based method. So we can do the first step of normalization. Again, we normalize the feature data first. Next, we normalize the data for the negative probes. So we can use the first step of normalization, log normalization, to visualize the matrix data for gene expression. The second step of normalization is Pearson correlation normalization method. So this step data normalization is a better method to cluster the cells. It is an improved method for both highly variable features detection and PCA generation. In the second step of data normalization, we will perform the Pearson correlation normalization method. But here we only normalize the gene data. We don't normalize the negative probes because we only use the gene data to cluster the cells. Let's perform the second step of data normalization. So we normalize the data by both log normalization and Pearson normalization method. Now we can add the statistics into the object. First, we add the statistics for the gene data. Next, we add the statistics for the negative probes data. Now the data is ready for dimensional reduction analysis. In this dataset, we are going to identify the highly variable genes. So we can use the calculator HVF function to identify the highly variable genes for the PCA analysis. Let's run. So you can see we identified the highly variable genes for the PCA analysis. So now we can use the highly variable features to perform PCA analysis. You can see here, for both calculate highly variable features and run PCA, we use the value from the Pearson data normalization. Because the Pearson normalization is an improved method for highly variable features identification and PCA analysis. So now we can run PCA. So after PCA, we can generate a scree plot to help identify the best PCA for UMAP analysis. You can see here from the histogram plot, from PC1 to PC4, we capture the most biological information. So once the PC reaches to PC8, you can see the curve is almost flat. If you add more PC numbers, probably not going to give you too much information. So the online tutorial uses the PC1 to PC10. We can also use PC1 to PC10 in the UMAP analysis. So let's run UMAP now. So we performed both PCA and UMAP dimensional reduction analysis. Now the data is ready for cell clustering. In the Giotto package, we run the first step to create the shared nearest network for cell clustering. So following the network analysis, we can identify the cell clusters. For today's demonstration, I'm going to use the Nathan method to identify the cell clusters. And you can see here, I followed the online tutorial. We use resolution as 0.07, but here I'm creating the cell clusters for the full dataset because we are analyzing the data for all the field views from field view 1 to 28. Okay, we identified all the cell clusters. Now we can visualize the cell clusters using the plot functions. First, we can plot the UMAP for the cell clusters. If you look at the cell metadata, the cell clusters should be in the latent cluster column. So we can set the cell color argument as latent clusters and plot the UMAP. Let's run. You can see from the UMAP, we have 15 cell clusters here for the full dataset. So now we can visualize the cluster on the spatial space. We can use the spatial plot function to see the cell clusters on the tissue. Let's run. We can zoom in. You can see now from the cluster color, you can see the localization for different cell clusters on the spatial image. Because we have a large data set here, it's very hard to see the individual cells at this magnification level. If you want, you can subset the image, use the x and y coordinates information here. Then you can visualize the subsetted image. So for example, we can subset the image by the position for the x from 3000 to 1000. For the y position, it's minus 157,800 to minus 159,800. So let's subset the field view first. You can see we named the new image as small field view. Here we only subset the coordinates because we want to visualize the cells in the subsetted image. So also we need to subset the feature IDs for the small field view object. Let's subset the feature IDs in this area. Now we can visualize the cells in more details using the small field view object. First, we can use the spatial in situ plot point function to look at the polygon outlines. Let's run. And we can zoom in. You can see the cell boundaries in this area we subsetted from the whole image. And also you can use the different code to look at the cell clusters on the subsetted small field view. We can run this code. Let's run. Let's zoom in again. So you can see on this small area, we only have 5 cell types from the UMAP cell canisters. They are labeled by different cell colors. So this is for today's demonstration. You can have a look at the online tutorial because the online tutorial has more demonstration. Because we already analyzed the object, you can just use the analyzed Giotto object and run other code in the online tutorial. So I'm going to stop from here. I don't have time in this video to show you how to identify the mark genes for cell canisters and also annotate the cell canisters, but the online tutorial showed you how to do this. And also in my previous Giotto video tutorials, I showed you how to identify mark genes for different cell canisters and how to annotate the cell canisters. So this is today's demonstration. I hope this video tutorial can help your data analysis. And also if you watched my video tutorial using the SquidPy thread package to analyze the non-cancer dataset, then you can get an idea which package is the best package for your data analysis. So please subscribe my channel if you haven't to do so, and also share my videos with your friends. Thank you.